The thing that made Locke and Kant really smart people who weren't like contemporary psychologists is that, you can't blame them, they didn't have methods for testing their ideas. That is, there simply were no methods sitting around for testing their ideas. But thank heavens we live in the 21st century. And in fact, even since way back when, before you were born in the 20th century, there have been methods for testing these ideas. This is no longer the most sophisticated of methods. But we'll start here for a reason that will become clear within 5 minutes. It's something called the twin method, which we've seen mentioned in earlier parts of the course. The idea of the twin method, which will then harken back to, and just to preview the whole thing, recast or rephrase this nature versus nurture argument. The nature of the twin method is that you can compare two types of twins. So, as seen elsewhere in this course, there's two types of twins, monozygotic and dizygotic, or in normal everyday language, identical and fraternal twins. The monozygotic twins have, for all intents and purposes, the identical genetic material in the twin pair. It's not totally 100% genetically identical all the time, but for our purposes, let's say it is. And the DC twins have overlapping 50% genetic material, which means you can compare them. The existence of monozygotic and dizygotic twins is like as if, whoever created everything handed social scientists a naturally occurring experiment. If you ran an experiment, you might manipulate something where some people get 100% of some drug and other people get watered down 50% to see if the drug makes a difference at different levels. Nature has handed the world genetic manipulation, in which some people have 100% genetic overlap and some people have 50% genetic overlap. And so you can see if genetic amount of similarity makes a difference by comparing MZ to DZ twins. I won't work out the comparison here today, but the comparison is pretty straightforward. You get a whole bunch of MZ twins. What I'm doing with my arm is drawing a graph. And DZ twins to take psychological tests. Some measurement occurs, and you see that the correlation pattern of MZ twins is tighter. They're more similar than the dizygotic twins. If you do that, the arithmetic of the twin method yields something that's very straightforward and easy to understand with simple pie charts that I'll put up on the screen in a moment. A proportionate variance. People are not all the same if you measure their intelligence or their personality. They aren't all identical. If they get an intelligence test score, they don't all get a score of 100. Some people get an 80 and some people get a 120. The question is whether these differences in scores, that is, the variation in scores, is a reflection of genetics or the environment. Clearly, for some things, the influence is going to be almost entirely environmental. How much do you know about ice hockey? It's not going to be wired in, and if you grow up in northern USA or Canada, you're going to get a higher score than if you grow up in Chile, no matter what your genetics. Some things are going to be genetic. Height is heavily genetic. Fingerprint patterns and lines and ridges on fingerprints is essentially 100% genetic. So sometimes the pie chart will tilt towards genetics, in blue here, and sometimes it'll, which Kant would like. The Kant is more complex, but it's a psychology class, so we can gloss over the philosophical complexities. And sometimes you would get a pie chart in which everything other than the genetics accounts for most of the action. Or, as will be shown, you can just represent these percentages in a graph. But this is a sense in which thinking about variation of slices in a pie is good, even though I'm about to switch to a graph. So that's the idea. In principle, you can just go out and run some sort of study and see if the thing you're interested in is primarily genetic or primarily environmental. And then one of these two guys who died hundreds of years ago will be happy. As you can guess probably from that setup, the story is actually more complicated than that. Here is a particularly illustrative study by somebody named Eric Turkheimer and his colleagues et al. Done, well, in that year. That's some number of years before you watch this film. What they did is the following. They had a very large national sample of citizens of the US of A. Once you have a large sample of people, you end up with some of them being twins. In this study, there were 320 twin pairs, like it says on slides that you presumably can see. Some of them are MZ twins, some of them are DZ twins, so you can compare the degree of similarity among the MZ and DZ twins, and if they're equally similar, then the difference in genetics made no difference, and if the MZ twins are much more similar than the DZ twins, then genetics did something. What they did in this sample is to measure intelligence through standard intelligence tests administered to everybody. So they'll have a graph here, if you turn your head sideways, that's the percentage of variability in intelligence that's explained by genetics. So not everybody gets the same IQ score. It might be that if hypothetically the MZ twins are always really similar and the DZ twins differ a whole lot, then the genetic similarity accounted for the intelligence similarity, and there'll be a big bar here that goes way up to the top of the screen. The thing that makes this study interesting is that there's no bar. What happens is that they had the presence of mind to take note of the fact that some of the people in their sample of Americans were poor, by American standards at the very least, if not total worldwide standards, and other people lived in relative wealth. The reason this might be relevant to something like intelligence is that if you live in an environment that's wealthy, you've got a buffer against a lot of environmental factors that might exist and impair intellectual development in a poorer environment. The most obvious one is nutrition early in life. The brain needs a lot of energy early in life. A lot of brain development is impaired by a lack of good nutrition. In general, once you hit a middle-class American society, they usually manage to come up with pretty good nutrition for small kids. And that's less true, plus the other disturbances of life that can occur in poor neighborhoods. So what they do in this study is, instead of computing a number that answers the question, how much did genetics influence intelligence, they compute a bunch of numbers for each of a variety of gradations in economic well-being or socioeconomic status, and they get this. What happens is there is no singular answer to the question, to what degree is variability in intelligence due to genetics. Instead, if you imagine a pie chart, there's no chart. There's different charts for different levels of intelligence. The results are actually quite striking. If you live in, once you hit, a middle and up economic welfare neighborhood in the US of A, the difference is, if you imagine, are they genetic or environmental, the differences in intelligence are, in fact, almost entirely genetic. Everybody has a perfectly reasonable environment. You're in a setting in which it's pretty unlikely that there's going to be, mass lack of nutrition. There's fewer gangs disrupting life. The school systems will have electricity and plumbing. A bunch of normal stuff will happen that, in a sense, will allow people to realize their potentials, and therefore genetic potentials get played out. In poor neighborhoods, the role of genetics shrinks all the way down to essentially single-digit numbers of percentage. That is, most of the variability is a reflection in intelligence, is a reflection of whether the person lived in a biological environment that allowed for normal brain development, had a household environment in which they had early life developed reading and logical skills that allowed them to go to school, schools that were supportive of intellectual growth. And so there is no one answer. This is the first sense of there being some kind of interaction, and that's the theme of today. There being some kind of interaction between genetics and the environment. In retrospect, one didn't even need human beings. This is going to be from a book by the Harvard biologist Richard Lewontin showing plant heights. Here's a simple question that you wouldn't expect to see in a psychology class. To what extent are differences in height among plants due to genetics? You might kind of think, but hopefully you won't by the end of this film, that there's a, quote, gene for height, and individual differences in plant height are going to be determined by genetics. So here's a bunch of plants 1 through 7. They genetically differ, and here they are grown at a particular elevation somewhere, and plant 1 is taller than plant 7. In this little demo, the plant biologist took identical versions of the plants. You can learn about that in some biology class. You snip off some plant and plant it. I guess that's why they call it plants. And grows the same plants at medium and high elevations. So the plants reading up to down are the same plants. They're triplet plants. The triplet plants at the different elevations differ in their ranking of height rather dramatically. Plant 1 is the tallest plant in a low elevation. It's the second smallest, second lowest plant in the medium elevation. It pops back up at the high elevation. It's not even a linear relation between height of plant and elevation. If you did correlations across these things, the correlations would be kind of low. What's going on is that there's some kind of interaction of genetic mechanisms and environmental context, even in something as simple as the growth of the plant, which means now, even though this for plants is more of a question for another class, that whatever the genetic mechanisms are, they're not just spitting out an outcome. They're existing in an environmental context that causes the genetic mechanisms to do something. That something may differ by context and produces, in this case, different plants. Here's finally back to the world of humans. Human example, these guys are twins. They've been, for filming purposes, they've been sanitized. These guys are twins. One of them, the one who is much smaller than the other, they were adopted early in life. They grew up in different households. The household in which the poor, smaller gent grew up in had parental figures who were bad at parenting for apparently psychological reasons. The result of bad parenting, lack of nutrition early in life, is that these two people differ so strikingly that, except for some hair patterns, you might not even think they're related, yet they're testably identical twins. So there's some kind of lesson, but then there has to be some second lesson, because you need to explain the first lesson, that if you have a certain amount of genetic material and you're interested in an outcome, you can't necessarily get from point A to point B, because the outcome could differ as a function of the environment that's being experienced, and not just in an obvious way, like, you know, if there's no nutrition, the person dies, but in a somewhat more subtle way in which the outcomes or the phenotypes are not predictable from just the genetic material or the genotypes. The environment is doing something that's affecting what the genotype does. This raises the question of what these genetic environment interactions consist of. And when I used to teach this course back in the 20th century, everything would end now and it would be a good question. Now some miracle has occurred and everybody's figured it out. Sadly, those people were not primarily in psychology per se. They're in a subfield called epigenetics, but this is one of those fields, for any of you interested in psychology and biology, in which the distinctions blur and you're kind of in both fields at the same time, because epigenetics is the study of how something the psychologist has always been interested in, environmental factors, behavioral experience in the environment, alters, in this case, gene activity. If you haven't seen this topic before, you might wonder, hey, how would that work? And if you want to find out how that works, this is one of those cases where the brief film is useful.